Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta from IIT Delhi. Today we are going to talk on module Hypersensitivity-II and III parts from paper Immunology. It is known that for persons sensitive to penicillin, injection of penicillin may cause an anaphylactic shock and may be fatal. Hence, it must be emphasized that hypersensitivity type 1 can vary in response. To repeat, the immune response qualitatively is same. It is just that the animal may be more susceptible to an antigen. We will look at the clinical aspects of type 1 reactions. So the objectives of this module are to know about experiments which led to the realization that anaphylaxis and allergic reactions can become systemic to create an atopic individual. To understand how allergy becomes systemic to create an atopic individual. How food allergens can use gut as a gateway to development of systemic allergy, and how the clinical picture correlates with the IgE production and histamine release. To appreciate that, we still do not have a complete understanding of how environmental factors can combine with genetics to produce atopy. Finally, we will try to understand the molecular basis for various treatments of allergy. So the concept map essentially consists of anaphylaxis and anaphylactic shock, for which various experimental tests and techniques are used to look at. First is the wheel and flare test. Second is the Prosnitz and Kustener test. The third is the Schulze-Dale technique. And we will also look at the systemic allergy, which essentially has the following elements: mucosal membrane including gut assay, gate for allergens, its treatment, IgE level, the parameter of genetics, and environmental factors. And finally, we will talk of a concept which is called allergic breakthrough. The immune system offers protection or prophylaxis. For individuals who are allergic or hypersensitive to a particular antigen, the immune response is excessive. Such individuals are said to be in a state of hypersensitivity. As this response causes tissue damage, rather than prophylaxis, allergic responses are also called anaphylaxis, and type 1 hypersensitivity is called anaphylactic hypersensitivity. In different animals, different organs serve as experimental systems to demonstrate anaphylactic shock. In guinea pig, it's lung. For rat, it is intestine, heart, and pulmonary blood vessels in rabbits, hepatic veins in dogs, and lungs and larynx in humans. An injection of 1 milligram only of egg albumin in a guinea pig, followed by another after 2 or 3 weeks, is a good demonstration of systemic or generalized anaphylaxis. The guinea pig starts wheezing and dies of asphyxia within few minutes. Pathological examination shows intense constriction of bronchioles and bronchi, contraction of smooth muscles, dilation of capillaries. In certain humans, similar consequences may occur following a wasp or bee sting, or administration of adrenaline. Actually, the administration of adrenaline counters smooth muscle contraction and capillary dilation and prevents fatal consequences. Sir Henry Dale discovered that administration of histamine mimics results of systemic anaphylaxis. He also discovered that uterus from a sensitized guinea pig is histamine and contracts on being exposed to the antigen. This demonstration is called Schultz-Dale technique. It was also found that a serum from such an animal can sensitize the uterus of another normal guinea pig. This is how the phenomenon of allergy was discovered, and role of histamine, mast cells, etc. was ultimately discovered. Medical people still prefer the term anaphylaxis, and the sensitized animal is called atopic. It is only much later that people discovered that milder form of anaphylaxis occurs in response to wide variety of substances which were termed allergens. Let us try to delve deeper into why only some individuals are allergic to specific substances or are said to be atopic or sensitized. First, let us trace the origin of the various terms to bring clarity to this discussion. Atopy was first described in 1923 by Coco and Cook, and the term is used to describe clinical symptoms of type 1 hypersensitivity, which includes asthma, eczema, hay fever, and urticaria. By that time, it was clear that anaphylaxis in animals discovered by Portier and Richer in 1902 had the same origin as hay fever or asthma in humans. However, Coca and Portier believed that the allergic diseases in humans were different from anaphylaxis in animals. Human allergy was known to have some hereditary aspects. There were no corresponding data for anaphylaxis. Hence, Coca and Portier called these diseases atopic diseases. Medical books use the term atopic to denote that a particular disease has hypersensitivity as its origin. Now we know that atopy, anaphylaxis, allergy, type 1 hypersensitivity reactions are essentially same. Different terms are, however, sometimes preferred depending upon the context. Prosnitz and Kustner in 1921 found that allergy can be transferred passively by serum. Kustner, who was allergic to fish, injected his own serum into the skin of Prosnitz, who promptly developed strong skin symptoms. About 45 years later, Ishizaka's group showed the involvement of immunoglobulin E in allergy. In a multidisciplinary subject like immunology, sometimes it is confusing to find different terms in different books or papers for what appears to be same or similar phenomenon. In such cases, brief historical perspective, such as what we discussed here, are invaluable. Immunology is an integral part of clinical medical practice. Let us further look at atopy or sensitized state from a medical perspective. The passive mode of sensitization occurs as the serum of the sensitized animal contains IgE to the specific allergen. As this was discovered by Prosnitz and Kustner, the test is named after them. The serum from allergic individual is injected into the skin of a normal individual. In the next 1 or 2 days, IgE diffuses towards nearby mast cells. The injection site responds with a hive's reaction if the allergen is injected there again. This is called passive cutaneous anaphylaxis. This slide discusses the difference between cutaneous anaphylaxis versus systemic anaphylaxis. Intradermal injection of an allergen results in local anaphylaxis. The response is in the form of what is called wheal and flare. Wheal is edema. Edema is actually swelling due to serum into tissue as there is increase in permeability. Flare is erythema, which is redness due to blood vessel dilation. In many cases, the wheal and flare reaction, which occurs within minutes, can be followed by late phase reactions, which can start within hours and can last for 24 hours. The edema is more pronounced. Intravenous injection, on the other hand, results in distribution of allergen throughout the body and may cause systemic anaphylaxis. Allergens like pollens and mite antigens are colloidal suspensions in the air and enter the body through inhalation. Apart from the nasal tract and respiratory tract mucous membranes, eye is also the contact point. Prominent respiratory allergens are hay fever and asthma. These are essentially local allergic reactions. In hay fever, the local release of the vasoactive factors leads to irritation, increase in vascular permeability, leads to runny nose, tears, and inflammation of tissues. In the upper respiratory tract, the similar local reaction leads to bronchoconstriction and difficulty in breathing. Usually, inhalation is possible, but exhalation is difficult. This overinflation of lungs causes damage in air sacs. Let us look at the phenomenon of food allergy. In food allergy, the local allergic reactions in the intestinal walls cause smooth muscle constriction and build-up of fluids. Intense discomfort follows diarrhea. Essentially, allergen binds to specific IgE on mast cells in the GI tract to produce these local reactions. If the allergen enters the body when gut permeability changes due to release of mediators, the allergen IgE complex can reach elsewhere such as joints, skin or lungs. That is why asthma is not caused by inhalation of allergens only, but often due to food allergies. Eating an allergen can cause urticaria or may precipitate an asthma attack. Gut of a sensitized individual acts as a gate to allergens. This is confirmed by the fact that the oral intake of sodium chromoglycate after ingestion of an allergic food prevents asthma. Sodium chromoglycate is known to prevent degranulation of mast cells. The serum concentration in humans of IgE is about 100 IU per ml. 1 IU is about 2 nanogram, which is 10 to the power 5 less than serum IgG concentration of 10 milligram per ml. IgE is only 0.001% of the total Ig in serum. Higher levels of IgE indicates atopy. Genetic and environmental factors control clinical symptoms. So a normal IgE level does not exclude atopy. However, if worm infection is ruled out and IgE level is high, atopic allergy is strongly indicated. While positive skin correlates well with IgE level, neither correlates perfectly with the clinical symptoms of atopy. That is, neither high IgE level nor positive skin test necessarily guarantee that clinical symptoms of systemic anaphylaxis will be present. Tada's lab in early 1970s used rats immunized with DNP ascaris along with Bordetella pertussis as adjuvant. The rise in IgE level was enhanced with rats from which thymus was removed or destroyed. Passive transfer of thymocytes or spleen cells from another population of primed animals suppressed the IgE production. It was found that TS cells reduce IgE production but do not affect IgE and IgM levels. Curious enough, neonatal thymectomy led to loss of IgE production. It is now known that in both humans and other animals, Th2 cells which produce interleukin-4 are required for IgE production. Th1 cells producing IF and gamma inhibit IgE production. Th2 cells are stimulated by antigen processing cells to release interleukin-4, which induces isotype switch to produce IgE. Responding to allergen, Th2 can inhibit Th1 response by producing interleukin-10, which interferes with allergen presentation to Th1. Th2 can also inhibit IF and gamma production. The above insights have led us to understand the role the genetics may play in type 1 hypersensitivity. That is why some individuals respond differently from others. Studies with humans as well as other animals have been carried out. Let us see the role of genetics in IgE response. Mutations in mice that inactivate interleukin-4 gene lead to abolition of IgE production even after a nematode infection. Patients given interferon alpha show reduction in serum IgE level. Interferon alpha acts similar to interferon gamma but has fewer adverse effects. Th2 cells also produce interleukin-5, which causes B cells to produce IgA as a secretory antibody. IgA is known to stimulate eosinophil development and activation. Th2 cells produced interleukin-4 and interleukin-5, and that is the basis of eosinophilia, which is quite frequently associated with allergy. Eosinophil count is a clinical aid in diagnosis of infections. Animals differ in the amount of IgE they produce in response to identical exposure to allergens. There are strains of mice such as SJL which do not produce high level of IgE even under optimum protocols. Low responder SJAL mouse makes only IgG in response to a single large dose of antigen and makes no IgG or IgE when repeated low dosage of antigen are given. A high responder C3H strain mouse produces IgE with both kinds of exposure. The repeated low dosage injection mimics natural exposure. As early as 1920s, there was data that allergy is hereditary. Both genetic background and high level of IgE in serum are risk factors for atopy. While non-identical twins show concordance of atopy only slightly greater than general average, even identical twins show much less than 100% concordance. This clearly shows that heredity alone does not determine atopic state. Among the known environmental factors are: number 1, nutritional status of the individual; number 2, presence of underlying chronic infections; number 3, acute viral illness; and lastly, quantity of the exposure. If allergic persons are identified by skin tests with different allergens, those testing positive show higher frequency of HLA-B8 and HLA-DW3 but not of HLA-A1. Higher responsiveness also correlated well with higher levels of total IgE in individuals with HLA-B8. HLA-B8 also shows strong correlation with incidences of autoimmunity. It is suggested that association of HLA-B8 to hyperactivity is via depressed TS cell activity, as depressed TS is involved in IgE responses as well as autoimmunity. Mast cells between different species and from different sites in the same species differ in their morphology and functional behavior. Connective tissue mast cells stain with toluidine blue after formalin fixation of tissues. Mucosal mast cells do not show up well with this stain. Connective tissue mast cells are found around blood vessels throughout the body, contain large amount of histamine and heparin. Their degranulation is inhibited by sodium chromoglycate. Mucosal mast cells are present in gut and lung. These depend upon IL-3 and IL-4 for proliferation, and their number increases during parasitic infection of the gut. They also increase in ulcerative colitis and Crohn's disease. Their number, the CTMCs number, increases in the synovium of rheumatoid arthritis patients. It is believed that both cells arise from a common precursor cell and local environment dictate their differentiation. Tryptase and chymase are two neutral proteases which are found in mast cells in different ratios depending upon the tissue. These proteases are of clinical interest. Tryptase is associated with bronchial hyperresponse, and chymase controls bronchial mucus secretion in asthma. Both proteases can degrade vasoactive intestinal peptide which mediates bronchial relaxation. Mast cells are seen to infiltrate the local sites in patients. Patients with hay fever have MMCs collecting in nasal epithelium during pollen season. In asthma patients, higher number of mast cells is seen in bronchoalveolar lavage fluid. Mucosal surface is the first site of contact between allergen and these superficial mast cells. This leads to release of mediators that increases the permeability of mucosa to allergen. Allergen now contacts submucosal mast cells which now release mediators. This is why clinical symptoms amplify. IgE binds to mast cells and basophils via high affinity Fc epsilon R1 receptor. This receptor is a member of IgE supergene family and is called classical receptor. Leukocytes and lymphocytes have another receptor for Fc of IgE. This is a low affinity receptor and its structure is homologous to animal lectins. Fc epsilon R1 is a tetramer with alpha beta 2 gamma composition. It has alpha chain, which is a glycoprotein and is involved in binding of IgE. The beta chain is linked to gamma chain via disulfide bridges. The binding constant of IgE with this receptor is 10 to the power 10 molar inverse. It is only the cross-linking of several IgE cross-linked on the surface which triggers degranulation of the cell. Fc epsilon R2, also known as CD23, is a 45 kilodalton membrane bound protein and occurs in two forms. The two forms 2A and 2B differ only in the cytoplasmic part, which is the N-terminal. 2A is generally expressed in B cells and 2B is expressed on T cells. B cells, monocytes and eosinophil depend upon IL-4 for induction. The 2B positive lymphocytes and monocytes increase in circulation of some atopics, especially atopic eczema. In hay fever patients, both IgE level and expression of 2B on lymphocytes increases simultaneously during pollen season. Normal alveolar macrophages which express 2B receptor in the presence of IgE positive allergen release enzymes which suggest their involvement in asthma or alveolitis. Number of compounds that can activate mast cells directly. These include calcium ionophores, melatonin, codeine and morphine. The common feature in their effect is calcium ion influx into mast cells, both direct triggering and via Fc epsilon R1 mediated triggering is illustrated in this slide. We are now ready to look at the various factors involved in the development of allergy. The integrated picture had led to the concept of allergic breakthrough. Finally, we will look at some possibilities regarding treatment of allergy. By now, it should be clear that allergy is a generic term. All allergies have some common features, yet each is distinct in its own way. The hypothesis of allergic breakthrough is just about recognition that clinical symptoms of allergy arise when overall immunological activity crosses a certain point, which is called allergic breakthrough. So everybody responds to an antigen. This response, once it exceeds this point, we tend to identify it as an allergy. Even genetically predisposed high IgE responder needs a sum total of concomitant factors which can be denoted by X, consisting of decreased TS cell activity, transient IgA deficiency or upper respiratory tract viral infections. Atopics during upper respiratory tract virus infection display aggravation of asthma. Basophils release histamine if anti-IgE is given. It cross-links IgE bound via Fc receptors. Herpes simplex virus, live or killed, enhanced histamine release. Interferons may mediate this. Interferon itself has the same effect as the viral infection. Injection of increasing doses of allergen often benefits. Increase in specific Ig and TS cell activity and decrease in specific Ig is seen in serum. Specific IgG presumably blocks specific Ig mediated responses. In normal rats and mice with ovalbumin as antigen, hypersensitization resulted in decrease in IgE response, including development of tolerance. High IgE responder did not develop tolerance. In their case, antigen-specific TS cells and their IgE-mediated responses did not change. We do not have clear understanding of how B epsilon memory cells are maintained. There are possibilities that dendritic cells continue to act as APC or idiotypic network system ensures it. Beta adrenoreceptor stimulants such as adrenaline and isoprenaline salbutamol, which causes bronchodilation, alpha adrenoreceptor inhibitors such as methoxamine and phenylephrine, all have been tried to control allergic reactions. Antihistamines such as pyralamine, promethazine, diphenylhydramine inhibit histamine, which is just one mediator. Tryptamine antagonist cyproheptadine blocks receptors for serotonin and histamine and may give relief with some patients. Anti-inflammatory drugs such as salicylates, phenylbutazones, and glucocorticosteroids work but are immunosuppressants. To summarize the lecture, we learned that atopy, anaphylaxis, and allergy turn out to be same phenomenon. We saw the difference between cutaneous and systemic anaphylaxis. We also learned how gut can be a gateway to allergens. We also saw the importance of IgE level and regulation of its production, the role of genetics and environmental factors in IgE production, and the role of allergic breakthrough. We learned the controlling of allergy. IgE is believed to have evolved as a defense mechanism against parasitic worm infection, which affect about 1/3 of world population. Allergy, anaphylaxis, or hypersensitivity type reactions shown by atopic individuals show that these individuals are caught in this crossfire as they are more vulnerable for reasons not yet completely understood, but are definitely complex and consist of multiple factors. Thank you.